abstracts, and all based on their graduate work. And we're over the overall, so they're all finalists, and the overall award is to recognize the quality and creativity of the project as well as the presentation. And with that, if our first speaker, Joshua Justison, would like to go ahead and get your starting slide up there. Also, at the time for questions, there's a Q&A button down at the bottom, which is the best way to put your questions in. Also the chat, and if you want to ask it yourself, raise your hand. With that, we'll go ahead and get started. Josh.
All right. Yeah, thank you. And so today I'm going to be talking about a comparative method that considers the various paths found within reticulate phylogenies. And so first, we might notice that trait evolution models, they're going to be central to understanding patterns in morphological diversity. These can be used to assess things like tempo and mode of evolution, and ultimately poke at questions related to adaptation or selection. And with an increased ability to not only detect, but also infer hybridization and gene flow, it's being increasingly recognized that gene flow can play a role in shaping patterns in trait evolution. And this challenges ideas of trait evolution where traits are controlled by a single species tree. And recently, there's been a slew of methods that can try to account for the mosaic of gene tree histories that might control trait evolution, whether that's due to gene flow or things like incomplete lineage sorting. And that's primarily what I want to talk with you all about today, is our contributions to this space, and then also comparing and contrasting various models of trait evolution. And so first, we might want to think about how we might model trait evolution. And continuous traits are often modeled with Brownian motion. This can act as a null model for trait evolution. And when it comes to the distribution of traits, we can think of that as a multivariate normal distribution with these associated parameters that represent the starting trait value at the root of the phylogeny, the evolutionary rate, and then lastly, the covariance matrix that represents the shared ancestry between species. And when I talk about various trait evolution models, I'm going to be primarily focusing on how this covariance matrix is computed. And so the lineage sorting model that we introduce is, I like to think of it as a hybrid of network and gene level evolution models. And I'm going to talk a little bit about both of these models and then kind of talk about how this is a hybrid between these two approaches. So first, we might consider trait evolution on a network, which is a model that can account for gene flow. And the way that this model works is it considers all of the various paths that lineages might travel along, and it considers the covariance of between these lineages along all of these paths. And then it considers the probability that lineages are going to travel along these paths. And these probabilities are dictated by the inheritance probabilities of the network. And these represent the proportion of genomic material or genetic material that is contributed from each parental lineage. And a couple things we might notice is that in this model, covariance is essentially a weighted average along all of these paths where the weights are the probabilities. It gets a little bit tricky when we're considering the variance. There's a reduced variance in hybrid descendants, and I'll talk a little bit more about this property when we talk about results, but just note that it's a thing. The other thing we might notice is that the weights are going to be assuming that lineages sort independently of one another. So if we have two lineages here to compute the probability of that path, it's effectively assuming that these lineages are independent at the time of that reticulation. However, this might not always be the case. When we consider things like incomplete lineage sorting or the multi-species coalescent, we might consider lineages coalescing prior to that hybridization. And in that case, they're not going to be sorting independently, and this might affect the weights of things. And this is where we can draw on inspiration from the quantitative trait model of Hibbins et al. That uses gene tree level histories to account for discordance directly. And the way that they compute their covariances is they consider if the purple are gene tree histories, is they're essentially computing an average across all of the various gene tree histories. And so our approach here, it's going to consider both of these things at various stages. So first, if we call these various paths lineage sortings, these are going to use the lineage sortings and covariances at the species level or at the network level. And then it's going to be weighted by the lineage sortings found at the gene level. And what I mean by this is each gene is going to be associated with a lineage sorting. So these first two genes right here, they're both going to have this same lineage sorting because they both go left at the hybridization. And so what we can do is we can count up the proportion of genes with a given lineage sorting, and we can effectively use these as our weights when we are computing the covariance. And one other thing we can do is we can use the multi-species network coalescent to make predictions about what these lineage sorting weights might be. So how we can think about the multi-species network coalescent is essentially a distribution for gene tree histories that's going to be controlled by the effective population size. And that's going to effectively determine when on average lineages are going to coalesce. And so we can consider a small population size where lineages are going to tend to coalesce very early, and a large one where they're not going to coalesce prior to this hybridization event. And effectively what this does is this is going to change around the weighting of these various lineage sortings. And so what we can do is we can input a given effective population size and compute what we would expect the weightings to be. And this creates kind of a flexible model that allows you to use different weightings to test various hypotheses. So for example, what we might expect under the multi-species network coalescent might differ from what we observe when we're actually counting the proportions of genes that follow each of these paths. And alternatively, we might be able to incorporate outside information. So for example, if we have gene ontology, maybe we're trying to measure or maybe our trait of interest is something like tooth length or limb length. We might hypothesize that genes that are associated with carnivory might play a role in controlling this trait. And so we might use the lineage sortings from genes that are going to be related to carnivory to make a new weighting and kind of test this hypothesis of how much these genes might affect this trait. And so we have a flexible model here that considers the various ways lineages can sort along the network. And now we want to compare this model against some other ones and assess performance. So what we did is we did a simulation study where we simulated under 4 different models of trait evolution. I'm only going to be talking about 2 of them. And what we did is we simulated under all of these models, and then we did this under a variety of different phylogenetic networks. So the first simulation model we did was under assuming the network trait evolution, where we varied the inheritance proportions. We assumed an evolutionary rate of 3, and we did this on the variety of networks. I'll be primarily focusing on the results of the first, but we fit various models. First, we fit the major tree, which we can think of as the backbone tree of this network. We fit the network itself, which is the true model. And then we fit our lineage sorting model using expectations from the multi-species network coalescent for a small, large, and intermediate population size, or effective population size. And what we found here is that incorrect models tended to underestimate the rate. First, we can notice that the network was able to recover the true evolutionary rate, but everything else tended to underestimate it. And that happened to be due, or we primarily attributed that to the way this reduced covariance property of the network model. And so if we're looking at the covariance matrix of the network model, we can notice these 4 cells along the diagonal here. These represent the reduced variance of hybrid descendants. And so we can contrast that with this model that actually performed the worst, our lineage sorting model, when we're assuming a very large population size. It actually only differs by these 4 cells. It doesn't have that reduced variance property. Another thing we can notice here is that in this case, the tree model was the least biased, and this might be kind of a comforting result. We don't always have networks, so sometimes we have to fit tree models to trait evolution. However, that comfort should be tempered a little bit because this seems like it's context dependent. The tree model was actually the most biased depending on which network we were using. Another thing we can notice is that for really relatively high inheritance proportions, that all of our models tended to converge at the true evolutionary rate. And this would suggest that when we have relatively high inheritance proportions, that even if we misspecify the model, we might be robust to some of these model misspecifications. And so tree models might be adequate under certain conditions, particularly when we have high inheritance proportions and for specific topologies. And this can be really nice and comforting because not all comparative methods can accommodate reticulation. For example, when these folks were looking at monitor lizards, they conducted both tree-based and network-based analyses. And I would argue that their tree-based analyses are still relatively reliable, considering that the network that they inferred has relatively high inheritance proportions. So the other simulation model that I wanted to talk about is simulating under our lineage sorting model where the weights are from the multi-species network coalescent. And so here we varied our effective population sizes, but kept our inheritance proportions equal. And now our true model in this case is the lineage sorting model where they're using the true value of the effective population size. And so what we can see is that there's going to be a bias when we're not accounting for population size. So both the network model and the tree model are always going to overestimate the evolutionary rate. They don't account for population size at all, and it's going to become particularly bad when the true population size is large. Alternatively, for our lineage sorting models, they did assuming the wrong population size tended to lead to bias. So assuming a small population size when the actual population size was large, or assuming a large population size when the actual population size was small, ended up leading to biases. Another thing we might note is that generally for small population sizes, again, we might have some robustness to model misspecifications. And this could be kind of nice if we consider specific types of gene flow. So for example, cases of hybrid speciation likely arise from few founders, which is going to, might imply a small population size. So not only the amount of gene flow, but the type of gene flow we might want to be consider when we're fitting various models and asking whether model misspecification, if we're going to be robust to it or not. And so what we have is we have a comparative method that can account for the various ways that lineages might sort across the phylogenetic network. And we can test a variety of hypotheses from this, and it can be readily applied to a variety of other comparative analyses. Since we have the covariance matrix, we can apply this to things like phylogenetic signal, phylogenetic regression, or phylogenetic PCA. And I have this implemented currently as a fork of phylonetworks. And if you want to take a look at that fork, you can scan this QR code. Otherwise, it's available on my GitHub. I'd like to acknowledge the many members of the Heath Lab, both past and present, as well as my committee. And thank you for your time. Yeah, if you have any questions, yeah, feel free to ask. And if you can scan the QR codes for both the fork of phylonetworks and the citations that I have.
Great. And there's a hand up. I've asked you to unmute and ask your question.
Hi, thank you very much for your talk. It was really interesting. And I was wondering, when you are like getting evolutionary rate parameters with a Bayesian tree, and you like get the sigma squared for a sample from the posterior distribution, do you think is that like form to accounting for this uncertainty, or is better like to try to use phylo networks in like clades with high hybridization rates?
Yeah, I might suspect that the uncertainty in phylogenetic are you saying phylogenetic trees, like the estimates between those? I might suspect that it might capture some uncertainty. I think it's still a little open-ended as to whether, because I guess we might suspect that if we're inferring a tree when it's actually a network, that there's going to be some bias in the tree estimation itself. It might not accurately capture that backbone tree. And I think that's still an open question as to whether that biases in the tree estimates are going to lead to downstream biases, or whether it kind of captures the macroevolutionary trait evolution signal. I think that would be an interesting thing to still look at.
Okay, thank you very much.
Thanks. And we should swap over the slides now if you could stop sharing. And if someone has a really brief question while we get the next talk set up. Up. Heather, we see you're at, yep.
Yep. Is it work?
Okay, so we will move on to the second talk in this session by Heru Handika.
Hi, everyone. Thank you for the opportunity to present in this symposium. First of all, I want to say to start with, it is important for us to improve the diversity and equity and inclusion in our field, but we rarely talk about to improve DEI, we also need to improve the accessibility and inclusivity of the software that we run our analysis on. And I think all of you aware that sequencing cost is declining, but the software that we needed to run the analysis is also becoming more complex. It's often requires significant investment for the computing resources and technical support, which out of reach for many people. And in general, it's requires significant bioinformatics skills, and some software have problem with memory management and also dependency issues. Docker helps solve dependency issues, but Docker is commercial software. It used to be free for everyone. Now it's only free for people with for personal use, research, and education. And many, and we don't know how long they will keep it this way. And many software only doesn't work on Windows. You may say, but Windows can run Linux software. But Windows subsystem for Linux also have problem with RAM and storage access. We can find a workaround for this, but if you all aware, this system also another layer of abstraction from the hardware, which make it, if we are dealing with data intensive application, we need to remove as many indirect access to the hardware as possible. And to illustrate this further, I'm Indonesian, I'm a mammologist, so I'm showing you here the statistic for mammal. Indonesia is the country with the most mammal in the world. And systematic biologists or evolutionary biologists in general run the analysis on their personal computers, and many of those are Windows. So how do we improve the accessibility and inclusivity of the software that we run our analysis on? So we need phylogenomic software that resemble day-to-day computing. So how do we do that? So we need to rethink the way we develop software. So for starter, we look for the best programming language for the task at hand. So we were considering Julia and Go at first, but as you can see in the statistic on the left, we were fall in love with Rust and Dart, mostly because Rust is high performance, is also memory efficient, which is important when you're dealing with big data set. And it's cross-platform. It has a modern tooling that simplify the development process. And Dart and Flutter is the most common programming language and framework for developing cross-platform GUI software. And it's also give access to Google Material Design that help us simplify developing accessibility features for the GUI application. So we developed SIGUL, which is short for Sequence and Genomic Utilities. So SIGUL was just published last month. It's available as single executable command line application. It's also available as graphical user interface application, also as library for Rust and higher level language such as R and Python. And with SIGUL, we target this part of the phylogenomic workflow, especially if you're doing target capture sequencings. We chose this because first, this like a high performance solution. And we also want to avoid developing novel methods, so we can focus on engineering the software. We want software that run on many platforms to the computer that most people have access to. So it's a practical solution, but also we hope it to be a proof of concept that this kind of software can be done. So in general, the design is like this. So the core library and command line application is written in Rust. We also wrote the GUI software where the graphical user interface is written in Dart and Flutter, and we use this amazing Flutter Rust Bridge library that provide the binding layer. It's basically help the Dart code communicate with the Rust code. We use similar approach for developing the Python library. So in general, Sigul basically run from smartphone to high performance computing clusters. It support many operating system, and just for an example, for Android, it support over 14,000 device models. And we have a growing list of features. So as I mentioned previously, it's mostly for target captures, but we also have been adding features for that can be used for whole genome sequencing data sets, for example, for raw rates and contiguous sequence statistics. And the command line application, we wanted to keep it simple, because the more complex you make your command line, the more chance that people's gonna type, the more errors gonna happen when people type it, and some people not used to use the tab in their keyboards. So with this simplification, it's help people that not used to using command line application. And even here, you don't even need to, for most tasks, you don't even need to input the file format. And for the GUI application, we also distributed through the official App Store, which not just simplify research, but also simplify teaching. So it's only one-click installation. And this method also help us to, because by distributing to the official App Store, it's also improved the quality of the app, because there are people that review the application from the store, and also additional check for the accessibility, and also improve the security in general. And for the Python application, you don't need Rust, the Python library, you don't need Rust knowledge for this. It's simplified dependency management, and it's available in PyP. So it's just pip install PySigul. And here on the right, I'm showing you a script, Python script that take, in total, this is less than 15 lines of code to concat an alignment. So these eliminate this kind of problem that you often see in the phylogenomic software, where the wrapper program, the interface is written in Python or R, whereas the actual data analysis is happening with the command line application, usually in C, C++, or Java. But with this kind of approach, you need to distribute to Conda, and maybe also you need to through a BioConda, and then when the new hardware arrives, it doesn't support, and then you become a dependency problem. So with PySigul, it's only pip install PySigul, and you can distribute the software through pip as well. So how does the performance of this software compare to other applications? So we compare here with AMAS and GoAlign. This is the program that we are aware is one of the fastest for this kind of analysis. So we track the memory usage, execution time, and core usage. And in general, Sigul is faster and also more memory efficient than AMAS and GoAlign. And for summary statistics, the difference is huge. And Sigul produce more statistics than AMAS in this case, but we do have some limitation with the memory usage for alignment splitting in Nexus input. We are working to improve this. How about the GUI? So the GUI and the command line, when you're running it, when we tested it in the same hardware, it's basically identical. Among 5 that we tested, 3 is basically the same, and the other 2 is somewhat different, but the difference is negligible. And here the video is showing you how do we do concatenate analysis on the desktop application. And this video is not edited; it's just as it is. And the desktop version also capable of handling large data. We tested it with whole genome sequencing reads of deer mice. The file size of this data set is more than 0.5 terabyte, and Sigul can handle that, whether you're running it in command line or in GUI. You see some differences here, but keep in mind that command line application also need terminal application to run, whereas the GUI software run on itself. And how about the Python API? So the Python we compare to BioPython and also to the command line version of Sigul, and we tested here for concatenate analysis on PyLucy, which that run BioPython in the background. You see the difference is very big, but for the Python API and the command line, the difference is basically negligible. And what are the challenges when developing this application? So, of course, the first problem is like, how do we support many platforms? So how do we make that easiest for us? So we a lot of the effort that we did was to make the release process automatic. So right now we use GitHub Action to release the software. In general, we don't need, in general, it's automatic, but only for the GUI software in an Apple, Microsoft, and Android that require manual release. And we do also have some problem with mobile operating system, because in mobile operating system, you're not allowed to access the file directly. So you need to move that file somewhere so that the program can access. It created a problem for us when inputting a large number of alignment. We have a workaround. It works when once it inputted, it fast, as you can see in the graph there. We are working on improving this aspect of mobile operating system so that the experience will be on par with the desktop application. How about improvement? So in general, now we have a system, a software that runs from smartphone to high performance computers. So the next step is for us is to try to start developing novel method. We have some ideas in mind, such as advanced alignment cleaning, and also trying to detect selective sweep, so that we can start working with the whole genome data. And we'll welcome for collaboration and also other, if you have some feedback about this. And this system we also have been implemented to NAPU, which is a field digital catalog. The idea is that we want to make field specimen collecting people that are working at the museum to when they collect the specimen, it's more reproducible and accessible. But there are many aspects of phylogenomic workflow that need improvement in the software. So it needs everyone's to start investing and developing accessible and inclusive software, because I think that the increased data generation should not come at the expense of decreased complexity for the user. And as we have shown you with this presentation, that it's possible. And I think especially for some of us that work in a foreign country, that collaborate with people that don't have access to computational powers, we don't have to keep asking ourselves whether the analysis we run can also be reproducible for them. And with that, I will summarize. SIGUL runs from smartphone to high performance computing clusters, and also provide API for Python developers. And the performance is consistent regardless of which version of SIGUL you use. We do have some limitation with mobile operating system, but we are working on improving on that. And with that, I would like to thank all of these people. Without them, SIGUL won't be the way it is today. And I will take any question and also your feedback. Thank you.
Okay, good. We've got a little bit of time for questions. And there's one in the Q&A from Mark Hibbins. In software development for bioinformatics, he often finds that there's a trade-off between ease of use and its power or flexibility. And so did you encounter or consider this sort of trade-off when developing SIGUL?
So yeah, let me read it again. Okay, yep. Yeah, there is a trade-off, I think, with the flexibility, and I think there's also a problem with Rust is the learning curve is more difficult, especially if you compare to like writing a Python code. But with the GUI, what we found, because the actual software the run is running in Rust code, it doesn't really show any much of difference in the performance, whether you run in GUI or command line application, as we sound. The trade-off that we have found is that writing Rust code is more, you need more code than writing Python, but the investment is only in short time like that. In long term, we found that writing Rust code is debugging Rust code is a lot easier than debugging Python. That's like the trade-off that often we have to deal with. But with the GUI versus command line, it's basically not much different.
Okay.
Thank you.
And we should have time for one more question, probably, if anyone has one. Well, I'll ask one as we go ahead and swap over slides for the next talk. So have you seen this being adopted, or is it just, it's out there too early? Because it just came out, or are you seeing that it is getting adopted all over the world?
Yes, there have been people using it. We saw some downloads increasing recently, and also people that are listed here have been using it since the beta version for their it already published in the publication. You can see in the citation there are people already use Seagull in publication. But the Python that we just released recently already seeing 1600 download for the PIP. We don't know people use it for their publication, but we did see that people already use it.
And a final comment slash suggestion slash question. Have you thought of outreach activities to popularize this in the global South? This is from Manasi Weratunga, and says that one of the biggest challenges is just getting the information to reach that part of the world.
Yes, we hope we can do more, because I have friends from a bunch of different countries here at LSU that help to spread the word. But I did also in Indonesia, especially, we did workshop. It's been a few times we did this to use Seagull as part of the pipeline. And last time we were able to teach people to do the full analysis of sequence captures with 12 hours of workshop. And it was a great help for us. So hopefully, yeah, please also helps spread the word if you are here.
Okay, thank you very much. With that, we will move on to the third talk in this session. This is by Björn Kopperud.
Thank you for the introduction. Hello, everyone. My name is Björn Kopperud, and I'm a PhD student in Munich, Germany, under the supervision of Sebastian Höhner. And today I would like to present my work about phylogenetic estimation of shifts in the tempo of origination. So in evolutionary biology, we see that species richness can be very different among clades. Some groups are very species poor, and some groups are species rich. For example, if you look at the ginkgo tree, it's represented only by one species, whereas its sister clades, the cycads, has several hundred species. And similarly, there are only a handful of coelacanths and lungfishes, but their sister clade, the tetrapods, is immensely successful with over 30,000 living species. Now, these are quite famous and obvious examples, but the question that I'm interested in, are these differences an exception in evolution, or do we see these differences all over the tree of life? Now, I take a phylogenetic perspective for this question, and specifically I use a birth-death model, which has rate parameters that control how often speciation events and extinction events happen. And so if we take the standard birth-death model, it makes the assumption that the rates are equal across the entire phylogeny. And so one of our questions is it possible to extend this model? And is it possible to tell if some clades branch at a different tempo than others? And it turns out that yes, it is possible. And we developed a software called PESTO, which is an acronym for phylogenetic estimation of shifts in the tempo of origination. So if you run PESTO, you will get a result like this tree, where we painted the average speciation rate per branch with some color coding. And here, this is the phylogeny of the primates. We can see that there was a 50% increase in the speciation rate for the clade that leads up to the old world monkeys. And PESTO is implemented in the Julia programming language, which is as fast as C, but it's also dynamic, and it's as easy to use as R or Python. And if you want to test it out, you can go to this link. Maybe it's a bit quick, but I will also have the link later on. So one question is, why do we need another method for this? There are several methods available, for example, BAM and others more recently developed. Why would we not use one of those? Those methods are excellent and they do work, but they all use some form of simulations or Monte Carlo approach in the inference procedure. Be it Markov chain Monte Carlo, they might use data augmentation, or, for example, stochastic mapping of rates on the phylogeny. And this is all well and good, but what if you have a tree that looks like this? What if I have a tree that's very large, 20,000 taxa? The challenge with existing methods is that they can be painfully slow for phylogenies that are this large. You may have to wait months for the MCMC to finish. And even if it does finish, you're not guaranteed for the MCMC to have converged, or you might want to adjust your priors. And it's not very practical for the size. So what is different with our approach, Pesto? Pesto is equivalent to what's called the birth death shift model, which is implemented in RevBase. As long as we have the same set of priors and settings, then both the RevBase approach and Pesto will give you the same result. So here I have plotted the branch-specific speciation rates with both approaches, and each point in the graph represents a branch. And if the dots are on the diagonal line, then both approaches are identical, but they give the same result. This is again the primates tree, and the top right point cloud represents the old world monkeys, and the bottom left cloud represents the rest of the phylogeny. So this is the equality with previous methods. Now, what about the differences? The biggest difference is in the runtime. So here I plotted a log-log plot of the runtimes of analysis with different tree sizes. And if you want to use RevBase, you may be able to run a small analysis in a few days. However, if you want to run a 20,000 tip tree, you will need at least 4 or 6 months, maybe longer. That's quite a long time, and I am not that patient. So instead, I would use Pesto, which finishes in a much quicker time. It's on the order of seconds or a few minutes, and you can do this even on a personal computer or a laptop. It's relatively quick. So how do we get this to work? How does it really work? To estimate branch-specific rates in Pesto, we use the so-called algorithm that's called a backwards-forwards approach. So in step 1, which is represented by the green arrows, we calculate what is the probability density of observing the tree. And we do this by starting at the tips of the phylogeny, and then we iterate towards more older nodes in the tree, and then we reach the root of the tree. This is also called a post-order tree traversal. And in step 2, we do something similar. We calculate probability densities, but we do it in the reverse order. So we start at the root, then we go towards more recent nodes in the tree, and we end at the tips. So in step 1 and 2, we visit each node only twice. There's no MCMC. And it turns out this is sufficient to calculate the posterior state probability of any rate category for any time and for any branch. So then we can use this posterior state probabilities to calculate the estimate for the speciation rate, which essentially is some kind of weighted average across our rate categories. And it's also averaged across the branch time. We can also calculate the number of diversification rate shifts, which is slightly more involved. You have to solve this differential equation, but we can do that with numerical methods, and it's relatively straightforward. Now, the nice thing with Pesto is that I will show you the primates tree again. We don't only get diversification rate estimates for each branch, but we also get estimates for the number of rate shifts. We get the shift size of the rate shifts. In other words, how much did the rate shift change? What is a small change or a large change? We also get the shift location, or where on the phylogeny, which branch was it that where there was an event. And we can also calculate this statistical support for the shifts. So on the left side, I have a scatter plot where on the y-axis, it's the base factor, representing the relative support for that there was at least one shift per branch. And on the x-axis, it's the branch indices. So in this case, on the primates tree, we had strong support for there being at least one shift on one particular branch. And that was the branch that led to the evolution of the old world monkeys. So this much more efficient method allowed us to investigate among lineage rate variation in an extensive data set of empirical phylogenies. So here I have 11 of these data sets. And in the first column, you can see 3 phylogenies of aquatic animals. In the second column, I have terrestrial animals. Third column, I have flowering plants and ferns. And I also have a mushroom-forming fungi and lichen-forming fungi on the right side. Now, some of these phylogenies are quite large. They can have 10,000, 20,000, 30,000 tips, but we're still able to do the analysis in a reasonable amount of time. If you look at the y-axis, we see the number of diversification rate shifts. Now, notably, all of the phylogenies we investigated, we can see that there is evidence for some among lineage rate variation. On the x-axis, you can see that the rate shifts are partitioned or broken down by the size of the rate shifts. So most of the rate shifts are small, they have a minor effect, but there are also some rate shifts that have a larger effects. You can also see that some phylogenies, some showed an overall signal of acceleration or deceleration by looking at whether this distribution is skewed to the left or to the right of 0. So if you look in the top left, I don't know if you can see my cursor, but in the top left, I have the rayfield fishes, and they're skewed to the right, meaning that they show an acceleration in the tempo of diversification. If you look at the mammals, they are roughly balanced with an equal number of upward shifts and downward shifts. And if you look at the bird phylogeny, you see that there is a deceleration in the tempo. Now, we can also summarize all of this acceleration and deceleration in the complete set of phylogenies, and we do this by calculating what's called the shift magnitude. So here I have 45 phylogenies, and if we look at the overall pattern, was there an acceleration or deceleration in the phylogenies? And we find that there is a roughly equal amount. So roughly half of the phylogenies that we studied showed acceleration, half of them showed a deceleration, and there's no strong trend either way. We also found that this acceleration or deceleration was not time-related, meaning that old phylogenies and young phylogenies are equally as likely to be accelerating or decelerating. Now, we also investigated the tempo of rate shifts, or the shift rate, and this is a measure of how often the shifts in the diversification rate happen. And in short, we find that shifts happen more often in young phylogenies, and they happen more seldom in old phylogenies. And this difference is quite large. It's about 2 orders of magnitude between the youngest and the oldest phylogeny. Although there is a lot of also, so this is on average, but there is also a lot of deviation from the average. So a lot of dataset specific variation. So in summary, we developed a fast version of the birth death shift model. So if you have an exceptionally species-rich phylogeny, it is now feasible to analyze this using among these methods. So you can run it on your laptop, and in a few minutes, you start the analysis, you go get a coffee, you get back, and it's you finished, usually. So this efficiency allows us to broaden our taxonomic scope and learn more about the origin of biodiversity. And our main result is that there is pervasive evidence for among lineage heterogeneity and the tempo of diversification. So I want to thank you for listening. I want to thank the members of the HUNA lab in Munich who are on this picture. And if there is time for 1 or 2 questions, I would be glad to answer them.
Perfect timing. We've got already 2 questions in the Q&A. If you want to open, 4 questions, and have a look at the Q&A. And first question's about whether you've tested it on reasonably complex simulated trees.
We also have tested the performance of these models on simulated trees, which I didn't show in the talk because I thought there was not enough time for it, and it's relatively complicated. But I have a supplemental slide where I have some notes on this. Where is it? Maybe the main question the main answer to if it gives you the correct answer is that we see very few false negatives, but we also see a very high false negative rate. So if there was, for example, a shift in the true tree that led to an increase in extinctiation, then the clade went extinct, then we are unable to recover that. But if we inferred a shift in our inference procedure, then there's like a very, very, very high chance that there actually was a shift. So false positives are very low.
Okay, and let's move on to the next question, which is, does your method allow for, does it require ultrametric trees, and does it allow for extinct taxa?
So our method uses based on it's the birth death shift model, meaning that we need complete trees that are only extant samples. We do not have a parameter for fossilization. So no, you cannot have fossil trees in this approach, but theoretically we could extend it to also add another parameter for the fossilization rate. But I think that is quite a big step at this point. So I'm sorry to disappoint.
If you go ahead and unshare so the next person can
Yes.
get set up, and we'll take one more question from the Q&A. It's a question about linking of speciation and extinction rates, but your estimates often showed shifts in only one or the other. And how did you parse out those parameters? Except actually we're out of time.
All right.
So I will leave that to the chat Q&A conversation.
Okay, thank you.
And we will move on to a talk by Layman Ball on floral diversity.
Can everybody hear me? See the slides? Okay. Thank you guys for being here. Thanks for coming to my talk. My name's Lamone Ball, and I'm a fifth-year PhD candidate at Louisiana State University. And today I'll be talking about some of the research that I've done as part of my dissertation. So I study this really diverse tribe of plants in the coffee family called Hyliae. It includes 29 epiphytic species that are spread across 3 genera called Hylia, Cosmobuena, and Balmea. So epiphyte means that these are plants that grow on top of other plants, and that's usually large trees. The tribe is distributed throughout Central and South America, but species richness really peaks in Southern Central America. Species display hawk moth, hummingbird, and bat pollination syndromes, which are suites of flower traits that evolve in response to different pollinator groups. So hummingbird pollinated species tend to have orange or red flowers that are tube-shaped and odorless. Hawk moth pollinated flowers tend to be white, narrow tubular. They open during the nighttime, and they also produce a sweet odor at night. And bat pollinated flowers tend to be pale green to yellow, tubular to bell-shaped, and they produce more of a musty odor at night. And so besides their floral diversity, Hyliae also occur in a variety of habitats, but mostly they can be found in more low to mid-elevation wet forests. And so the group's distribution throughout Latin America and that high floral diversity raise really interesting questions about how the group evolved. And so some of the questions that I'm interested in are how these species are related to each other, first of all. How have geographic distribution, bioclimatic niche, and pollinator use evolved through time? How are these variables partitioned between different Hyliae species? And then what can these macroevolutionary patterns tell us about plant evolution in the neotropics more broadly? And so first, to understand those phylogenetic relationships in Hyliae, I had to get some genomic data. And so to do that, I decided to design a custom Ruby ACE specific probe set for target enrichment. And I did that using genomic resources that were all publicly accessible, as well as a few different bioinformatic tools. I tested the probe set using both simulated and real target enrichment data from 27 taxa across the family, which are included in this phylogeny over here. And if anybody is interested in the details of how I did that, please check out this paper and applications in plant sciences. There's a QR code there. And so this probe set design worked really well. We were able to get great sequencing data back for Hyliae. And so here is the Hyliae species tree that I inferred after wrangling all of that genomic data that I got with the probe set. We sampled about 83% of all species in the tribe and included representatives from all genera and subgenera. 1,375 filtered gene trees went into that astral species tree analysis. Here, the local posterior probabilities are shown below the branches. And for the most part, these are all really high, above 0.95 throughout most of the tree, which is great, especially considering that we did have quite a bit of gene tree discordance in our data set. Most of the phylogenetic uncertainty was coming from this group here within Hylia subgenus Hylia. And so to better understand some of the potential sources of gene tree discordance, I used this program called quartet sampling. And so this program calculates a few different quartet-based scores, and then you can use those to better understand whether gene tree discordance is coming more from a lack of information or support, or if there is evidence of incomplete lineage sorting or another biological process outside of incomplete lineage sorting causing discordance. And so the main thing to take away from these results is that we estimated the lowest quartet sampling support values also in that group, Hylia subgenus Hylia. And those low scores in the subgenus are consistent with discordance being due to biological processes beyond background lineage sorting, such as, for example, introgression. And introgression is a plausible scenario, especially because all of these species have that hawk moth pollination syndrome, and they all occur throughout the Andes, meaning that there could potentially be some gene flow between them. But other processes such as heterogeneity and evolutionary rates or base compositions could also be causing discordance. And some of my future work will focus on the phylogeography of this subgenus specifically, and testing for hybridization and regression to better tease apart some of these different potential sources of gene tree discordance. Next, I estimated divergence times for the species tree, and I performed a biogeographic reconstruction. So the 95% HPD for the node ages and the estimated ancestral ranges are shown on the nodes. And I estimated that Hylia originated in southern Central America about 19 million years ago. And since then, they have had this pretty dynamic biogeographic history characterized by frequent dispersal between Central and South America. And so altogether, there have been 4 independent dispersals from Southern Central America to Northern Central America, 5 from Southern Central America into the Andes, and then 6 from the Andes into the Amazon. And then once a lineage left from Southern Central America, we didn't see it return there. Next, I describe species bioclimatic niches using a principal components analysis of environmental rosters from the World Climate database. Before the analysis, I clipped the rosters so that they just covered Hylia's range. And so points in this PCA represent land pixels in each of the regions over here. And so the first 2 PC axes explained 82% of the variability in the environmental data, and those were most strongly associated with elevation and precipitation seasonality. And so here we can see that topographic and climatic diversity of the Andes in Northern Central America in light blue and yellow over here is a lot greater than in Southern Central America or the Amazon, which overlap here and take up a lot less space on the PCA. And so then to get the species bioclimatic niches from that data, I identified which land pixels in the PCA each species occurs in, and then I took the average score of those pixels on each PC axis, and then used that average score as a measure of niche identity. And so species niche identities are plotted over here, and they're color-coded according to their geographic ranges. And from this, we can see that most species cluster actually pretty closely together on this PCA, which suggests that they're really just using a subset of that total available niche space across their whole range. We also performed an L1OU analysis, which uses Ornstein-Uhlenbeck models to detect evolutionary shifts. And so using those first 2 PCA axes as our measure of bioclimatic niche, L1OU detected 2 significant niche shifts. So the first was in Balmea stormii, as it was dispersing from Southern Central America into Northern Central America, and also into drier, higher elevation environments that are more seasonal. L1OU also detected a shift in this Andean subclade within Hylia subgenus Hylia. And so that shift represents movement into wetter, higher elevation environments that are less seasonal. And so we see these two significant shifts in bioclimatic niche, but for the most part, niche has been pretty well conserved in hilly E. And that might reflect limits that are imposed potentially by their epiphytic habit. And so since these plants are getting all of their water from the surfaces they grow on, rather than the soil, they probably need to be in environments that get a lot of consistent rainfall. While Mayr seems to have been able to break out of that constraint, and it's the only species that grows in significantly drier environments. And interestingly, this is the only species that is variably terrestrial or epiphytic. And so a lot of the time, the species is actually growing from the soil. And that probably minimizes some of the water stress that species would be experiencing in these more seasonable environments. Okay, so next, I performed an ancestor state reconstruction of pollination syndromes. And so based on these results, we found that hilly E was likely ancestrally hawk moth pollinated. We detected 2 independent shifts from hawk moth to bat pollination, one from hawk moth to hummingbird, and then one from bat to hummingbird. So taking the biogeographic and pollination syndrome reconstruction results together, it seems like most of the pollination syndrome evolution has happened within Southern Central America, or sometime during species dispersal out of that region. And surprisingly, no pollinator shifts have occurred in this Andean subclade of Hylia subgenus Hylia, even though pollinator diversity is really high throughout the Andes, and there's especially high hummingbird diversity in those higher elevation environments. And so we have this case where all pollination syndromes are represented in Southern Central America, and multiple shifts between syndromes have happened there, whereas species that have diversified within the Andes have maintained that ancestral hawk moth syndrome. And so I wondered if this disparity in syndrome diversity might be related to species ecology, specifically whether higher competition between species in Southern Central America could be leading to more pollinator shifts in that region. So Southern Central America is incredibly biodiverse, and hilly E diversity is actually highest in that region. But at the same time, it's also relatively small, with much lower and less extensive topography compared to the Andes. There's also less climatic diversity in Southern Central America, which we saw in this PCA. I'm just comparing the 2 subregions there. And so it's possible that species there compete more for pollinator resources just because they're packed together more tightly. And so switching between pollination modes might be a way for them to facilitate coexistence. And so to get at that question a bit more, I calculated pairwise spatial overlap between species using Schoner's index of co-occurrence. And my expectation was that if there is more competition among those species in Southern Central America for pollinators, then those species should co-occur more often than in the Andes. And also that species with different pollination syndrome should co-occur more often than species that share syndromes. And so some of those patterns came out. Here are species that occur in Southern Central America, highlighted in red. And overall, pairwise spatial overlap was pretty high among those paired. And then species with different pollination syndromes do often overlap, but it isn't always the case. We saw that hawk moth syndrome species in Hylia subgenus Tetrandri frequently co-occur with each other, as well as with other Hyliae, regardless of syndrome. And so one of the reasons why I think that might be is because species in this group have really varying corolla lengths, going from as short as 25 millimeters in this Hylia palmana to as long as 75 in Hylia loranthroides. And that suggests that they might be using different species of hawk moths with different proboscis lengths, even though it's the same syndrome. And so during upcoming fieldwork, I plan to observe pollinators for a bunch of these species and collect floral trait data to better understand how pollinator niche has evolved and how these species are partitioning pollinator resources. And so something that I've taken away from this study is that it's not just one factor or another driving Hilly diversification, but it's a combination of both geographical factors and ecological ones shaping the group's evolution. And it seems that neotropical topography specifically plays a really important role in shaping the ecological context of flowering plant diversification. So with that, I would like to thank everyone on this slide, all of the Lago Marcino lab members, both current and past, for all of their help and support, as well as my funding sources on the bottom. Thank you.
Thank you. And we have one quick question in the Q&A. If you could go ahead and unshare, we'll get the next slides up. And Rosanna Zanil Ferguson is asking about the hosts of the lianas. Do the species prefer specific trees?
I'm not sure. I haven't seen these species enough in the field to get a sense for if they have like a taxonomic or group preference for certain trees, but it could be. So when I go and do field work in the next couple of months, I'll definitely pay attention to that.
Okay, great. With that, we'll move on to our next talk by Amanda Da Silva on mutualisms.
So hi, I'm Amanda, and today I'm going to talk a little bit about one of the directions that fascinates me, which are the mutualisms. So mutualisms are one of the main drivers of diversification. And when we think about mutualisms, we usually think about the benefits involved in this interaction. For instance, if you think about pollination, without flowers receiving pollen transfer and the pollinator receiving the nutritional reward. And we usually think about these interactions in a pairwise perspective, bee flower, aphid ants, but some organisms can interact with a lot of organisms at the same time. Plants, for instance, can interact with pollinators, and pollinators will gather the nutritional rewards, and the flower will receive the pollen transfer. But they can also interact with protective mutualists. For instance, the ants that patrol the plants and repel the herbivores. And this protection mutualism is mediated by the secretion of extrafloral nectar, which is a substance rich in carbohydrates. And here you can see the ants repelling a possible herbivore. So if they are really aggressive against herbivores, they can also be aggressive against pollinators, right? And this is called the ecological cost of one mutualist in another, the interference of one mutualistic partner on another one. In my first chapter of my thesis, I was trying to investigate the effects of ants on pollination. So here in the x-axis, I have the effects of ants on bees and butterflies, and in the y-axis, the effect size. The negative values represent the negative effects of ants on bees and butterflies. So reduction in the number of visits, for instance, and positive values indicate the positive effects of ants on pollinators. For example, more visits. And what we observed was that ants have a negative effect on bees, but they don't affect the butterflies. Here's an example of ants being annoying against bees. So they can repel also the pollinators and not only the herbivores. And if there is a conflict between ants and bees, it's possible that evolution has shaped some strategies that reduce this conflict. For instance, it's possible that protection mutualisms has a higher chance to evolve in plants not pollinated by bees. And one system in which plants are really high dependent on bees is the bus pollination system. So in this system, plants have this pericidal anthers. So the pollen are only released by bees that are able to vibrate the anthers. So the pollen is released by these apical pores. And if ants can affect the pollination only of bees, flowers with this pericidal anthers might be more affected. So the conflict should be more intense in these flowers than in flowers that have another type of anthers morphology. But most studies studying this interference of one mutualism in another or interference of one ecological interaction in another, they're usually from an ecological scale. And I think this is one of the first studies to investigate this from an evolutionary scale. So here we hypothesized that this Ant-bee conflict might constrain the simultaneous occurrence of traits mediating the protection mutualisms by ants and bus pollination in angiosperms. And the first step was trying to investigate which are the plants, the angiosperms. So I got all the angiosperms from the Plants of the World online. And I used the world list of plants with exophoronectaries from Marjorie Weber. And I did this study in genus level. And I consider that if at least one species has exophoronectaries, then the whole genus might have exophoronectaries, so the interaction with ants. And I got the bus pollination data set from this study from Avery Russell. And I also consider that if at least one species has parasital anthers, then the whole genus could have parasital anthers. And I used the phylogeny, the angiosperm phylogeny from Jensen's, and I tested the correlation, the evolutionary correlation between ant protection and bus pollination using Markov K-states model. Okay, so what did I found so far? So here we have on the x-axis plants that are not involved in any mutualisms at all, so no bus pollination and no interaction with ants. Plants involved in the mutualisms with ants, bus pollinated plants, so pollinated by bees, and plants, bus pollinated plants and involved in protection mutualisms with ants. And in the y-axis, I have the number of genera of plants involved in each one of these interactions. And what we saw was that mostly plant genera are not involved in any mutualisms at all. Only 6% of all plant genera are involved in protection mutualisms with ants, most of them from Fabaceae, Euphorbiaceae, and Bignoniaceae family. 5% of all plant genera are bus-pollinated plants, most of them Melostomataceae, Araceae, and Ericaceae. And only 45 genera, which represents 0.003% of all plant genera, are involved in protection mutualisms by ants and bus pollination. And most of these plants are in the Dipterocarpaceae, Fabaceae, and Polygalaceae family. And here we have the phylogenetic distributions of these two traits. So in the inner part of the circle, in light blue, there is the absence of exophoronectaries, so plants that are not involved in the protection mutualisms by ants. In dark blue, we have the plants with exophoronectaries, so plants involved in the mutualisms with ants. In the outer part of the circle, in yellow, plants with parasital anthers, so plants not pollinated by bees or pollinated by other types of pollinators, not all bees. And in orange, we have plants with parasital anthers, so plants highly dependent on bus pollination. So we can see here that most of the plants that have one trait, so if they have parasital anthers, they don't have the other trait, so they don't have the exophoronectaries traits. And as I said, I test this correlation between bus pollination and exophoronectaries using Markov K-states model. So we tested an independent model against a dependent model. So the independent model was the evolution of non-parasital to parasital and exophoronectaries from non-exophoronectaries. And the model that was favored was the dependent model. So there is a correlated evolution between these two traits. So here we have plants without these traits. So plants that are not bus pollinated and plants that don't have the exophoronectaries. So these are plants that are not involved in both mutualisms. and plants involved in mutualisms with ants. And we can see here that that's a chance of plants to gain the extrafloronectary traits, but there's a higher chance of plants to lose these traits. If you see the interaction from the plants that have parasitic enters, so plants, bus-pollinated plants, but plants that don't have extrafloronectaries, we also see that there's a chance of plants to evolve the parasitic enters trait, but it's more likely that plants to lose this trait. But we are interested in plants that have simultaneously involved in this two mutualism. So plants that have parasitic enters, bus-pollinated plants, and plants that have extrafloronectaries, so plants that are protected by ants. And we can see here that if plants have these two traits, it's highly likely that plants lose the parasitic anthers and not gain. So this seems that EFN, the evolution of extrafloronectaries, constrains the evolution of parasitic anthers. So the evolution of plants with parasitic anthers and extrafloronectaries should have occurred from plants with parasitic anthers. And so the evolution of parasitic anthers does not constrain the evolution of extrafloronectaries, as we can see here. So we corroborated our hypothesis that this conflict between ants and bees might constrain the simultaneous occurrence of traits mediating this protection by ants and bus pollination in angiosperms. And it seems that this happens because the ecological costs, especially the ones that compromise plant fitness, could be an important selective force, because plants that are involved in protection by ants might have their fitness reduced if ants repel the pollinators. And the ecological cost, the way we measure today, might be low, because these conflicts may have been minimized over evolutionary time. So for instance, plants with ants are not pollinated by bees. But one thing that got me thinking about this result was that some plants have both traits. So they have extrafloronectaries and parasitic anthers. But if this conflict is so intense, what happens to these plants? And we raised some hypotheses about this. I will talk about three hypotheses. The first one is that these plants might be less dependent on bee pollination. For instance, they might be self-compatible. Maybe the bus pollination is an efficient system, and even if there is a conflict between ants and bees, short visits could be enough to pollinate the flower and generate some seed set. And also maybe these traits arise in some environments, like the environments with a high herbivory pressure, but also with a high abundance of bees. And I'm going to study this hypothesis in the future. So it's going to be my next chapters. So I'm going to study how the arise of extrafloronectaries changes the floral phenotype in Chemicrysta, which is a bus-pollinated plant. And how does the herbivory gradient affect the evolution of extrafloronectaries and some flower phenotypes also in Chemicrysta? I want to thank you all for watching the presentation. And as the take-home messages, I want to say that there's a negative evolutionary correlation between traits that mediating this bus pollination and protection, but this might be common across other kinds of mutualisms, and that the ecological costs of mutualisms might be important for shaping the evolution of traits, especially the ones affecting fitness. So we should study more the mutualisms, not only from a pairwise perspective, but from all the interactions that happens at the same time. Yes, that's it. Thank you.
Okay, and we've got a couple of minutes for questions. The questions in the Q&A are still left over from the last talk, but. Okay, there's a question in the chat from Rosanna Zaniel Ferguson. Have you done the stochastic mapping of the two traits?
Yes, I didn't show it here, but I did.
And yeah, then if you look in the chat, you'll see that there's a bit more. So with the stochastic mapping, what did you find?
I can't see the one with the. I'm not sure if it's my internet
Yeah.
connection.
Yeah, so what Rosanna says is that they found evolving back to more non-porous sidality is faster than evolving to it. And so is it possible that during the time a lineage is porocidal, it's experiencing enough conflict that conflict is what's actually promoting the reversal?
Yeah, I think that was it. What's happened. I would need to check it again. I'm sorry.
No worries. Thank you very much. Did I miss any? Oh, one more quick question. Do you know what would happen if you move to a species level phylogeny? Recognizing there were good reasons to use the genus level phylogeny, basically, is that going to be an okay approximation, or?
Yeah, I use the genus level because there's a lot of plants that we don't have information about, and there's thousands of plants that I can't work on. But I'm going to use the species level in the future. I will select some species that we have data. I was thinking about a couple of them, but mostly Kamekrista, in which I can test this evolution quantitatively.
Thanks. We are out of time, but I'll point you to one more interesting question in the Q&A that you can answer in there. And with that, we will move on to the last talk in our Mayr Symposium by Jenna McCullough. Oh, Jenna, your slides were there just a second ago.
Sorry, I thought it was all ready. And of course, then
You were.
it goes out. Okay. Great. Can you hear me? I'm gonna assume so. Great. So thank you for everyone for coming today. I'm very excited to talk to you about my dissertation research on island kingfishers. So the paradox of the great speciators describes a pattern in which some species are spread across many island systems and show little phenotypic differentiation across their range, whereas others inhabit the same ranges, but each island has a phenotypically differentiated form. These are often considered subspecies in a single polytypic species complex. When Diamond and Mayr coined this paradox, they did not have a way to measure genetic distinctiveness of these great speciators. Therefore, phenotypic differentiation was used as a general metric for genetic differentiation. The paradox is, because they occur across many islands, these lineages have high dispersal ability. But since different islands have differentiated forms, they are likely genetically isolated. This would imply that they are poorly dispersing, and thus the paradox. I like to think of these three components of great speciators like a triangle, with dispersal ability, genetic differentiation, and phenotypic differentiation as these three key components for the paradox of great speciators. This paradox is based on geographic variation of Indo-Pacific birds, but not all groups of birds in this region are considered great speciators. And great speciators differ from other famous island endemic adaptive radiations because they involve rapid diversification without the evolution of obvious adaptive changes like bill morphology. One of the most prominent great speciators is the colored kingfisher complex, which was so important to Ernst Mayr's ideas that they were on the cover of his influential book, Birds of Northern Melanesia, in 2001. Previous work on the systematics of Todiramphus kingfishers found that this great speciator comprised upwards of 10 different species and highlighted rapid diversification, but did not find a well-supported relationships within this complex, or across the genus more broadly. One reason why could have been due to limited taxonomic or character sampling. Therefore, for this project, we investigated the genus's entire geographic variation and fine-scale patterns of trait evolution to delve deep into the processes underlying a genus of great speciator. For this presentation, I'll be discussing these three facets of great speciators. So theory predicts that island populations of a great speciator to be genetically and phenotypically differentiated and walk a fine line of intermediate dispersal ability. For this project, we did moderate coverage, whole genome resequencing for all named taxonomic diversity across Todiramphus kingfishers. The genus has 29 species, but there are 93 phenotypically distinct taxa comprised in multiple polytypic species complexes. From these data, we extracted 4 types of molecular markers. These datasets varied in the total amount of data from 13 mitochondrial genes and 11,000 base pairs to over 8,000 protein coding genes with 5 million amino acid sites. Across all datasets, we noted that the backbone of the Todi-Rampus tree is characterized by many short internodes, suggesting rapid and successive speciation events early in the evolution of this group. Instead of talking about the relationships, some biogeographic themes in this tree include species only found on a single island, continental species with no geographic variation, continental species with substantial geographic variation, species with geographic variation across continents and islands, species with geographic variation across only islands. Moreover, there's variation in migratory behavior, in which in a single species complex, we have a single widespread migratory taxon that breeds in Australia and migrates north with multiple peripheral isolates that have become sedentary. We have many cases of sympatry on islands, and we have cases of continental sympatry. So it's complex, but it's not just geographically complex, it's also phylogenetically complex. We found a high degree of congruence between datasets and tree building methods across most of the species with strong support, but some taxa, which are bolded here, either showed different relationships or lacked support. When comparing site and concordance factors across the 2 major topologies, we found that many of these very short internodes were approaching values consistent with high incomplete lineage sorting. Moreover, we found cases of mitochondrial discordance with several subspecies of Pacific kingfisher, sasser, having some or all of the sequenced individuals grouping with an entirely different species, sacred kingfisher, sanctus. When we explicitly tested for gene flow with ABA-BABA tests, we found that these 5 mitochondrial discordant sasser taxa have elevated F4 ratios with other sasser and sanctus, suggesting that these are hybrid lineages. Moreover, across the whole tree, we see signatures of ancient introgression between clades earlier in the evolution of the group, ranging from 2 to 37% of the sampled genome. Overall, this helps explain the topological incongruence between the different genetic datasets and tree building methods. So going back to our predictions, we can see that these island systems aren't as genetically isolated as we expected, and gene flow could have been present as these lineages colonized islands. Now, what about dispersal during island colonization? Dispersal ability lies at the heart of the great speciator paradox. Too much dispersal ability will contribute to pervasive gene flow, but too little dispersal ability inhibits colonization into islands. Therefore, it is hypothesized that intermediate dispersers will have the highest speciation rates. However, when empirical phylogenies of great speciators were used to test this hypothesis, they did not support this relationship. So to test this with Todi-Rampus, we quantified dispersal ability with the hand wing index, which is a measure of wing shape, with larger values of hand wing index indicating more dispersive wings and vice versa for lower values. We collected a fine scale dataset of all taxonomic diversity for nearly 1,000 specimens. So to test the relationship between diversification rate and dispersal ability, we produced a time calibrated tree using UCE data, secondary calibrations, and island ages in BEAST. We inferred that the most recent common ancestor for the genus is around 11 million years, and that most of the diversity in the genus has accrued in the last 3.5 million years. Here's an ancestral state reconstruction of dispersal ability across the tree, with purples being less dispersive morphologies and yellows being more dispersive morphologies. And we have these box plots that show taxon averages. We see that the most dispersive birds are those that undergo migration or have established seasonal movements, which is expected, and that the least dispersive groups include sedentary forest birds on New Guinea and those that colonized the most remote reaches of the South Pacific and French Polynesia. Now, it is expected that the sedentary forest birds would have the least dispersive wings, but it is paradoxical that the taxa that have dispersed to the furthest islands also have the least dispersive wing morphologies. When comparing estimated speciation rates and dispersal ability, we found that lineages with the intermediate values had the highest overall diversification rates compared to those with more extreme values. Though not perfect, this shows a similar bell curve as the simplified hypothesis that I discussed earlier. The French Polynesian birds are outliers to this pattern, We tested some explanatory variables, like island size and time since divergence, but they were not supported with phylogenetic linear models. However, I opted to leverage a famous pattern in declining land bird species richness in the Pacific. Each of these purple lines are the eastern edges of different avian families highlighted below. We can use this pattern as a metric for an island's isolation based on the number of land bird species present, with islands very close to New Guinea having more species than those in French Polynesia. So on the y-axis here, we have dispersal ability, and on the x-axis, we have increasing isolation. And what we see is that dispersal ability decreases with increasing isolation. In other words, taxa that inhabit the most remote islands, again, have the least dispersive wing shapes. This kind of relationship has been shown before with skeletal measurements, but it is, to my knowledge, the first time it has been shown with wing shape. This declining pattern of hand wing index and isolation suggests that wing shape is following the island syndrome. It means that predictable evolutionary trends are contributing to the erasure of the relationship between intermediate dispersal ability and speciation rate in island Todirampus. So going back to our predictions, we can see that lineages with intermediate dispersal abilities have higher rates of diversification, but that this relationship is being obscured by predictable evolutionary trends towards flightlessness for isolated island birds. Now, what about phenotypic differentiation? The defining characteristic of great speciators is their incredible phenotypic diversity across island systems. This has been focused on feather patterns and colors. Hypotheses for why color might evolve rapidly include sexual selection, in which more sexually dichromatic species evolve color faster, competition, in which sympatry between closely related species contributes to faster color evolution, and neutral processes, in which drift in small populations contributes to faster color evolution. To study this, we collected a large spectral reflectance dataset representing all Todirampus taxa across 15 plumage patches. Using these data, we can see that the genus takes up 65% of all volume in color space. And we can use XYZ coordinates and chromatic and achromatic measures of color to do multivariate analyses of these two aspects of color for both sexes. Here, purple are slower rates and yellow indicates faster rates. I'm showing male rates here, but we do show differences in rates between the sexes, and males did not always have the fastest rates of color evolution. We can also see differences within single species complexes, such as within Pacific kingfisher, sasser, and colored kingfisher, Flores. So what is the relationship between the rate of color evolution and lineage diversification? The y-axis is the rate of color change, and the x-axis is the lineage diversification rate. We can see that phylogenetic linear models indicate a positive relationship between the rate of color evolution and lineage diversification rate. This was true of both metrics that we use to calculate diversification, as well as for both chromatic and achromatic rates, and for both sexes. So in other words, more rapidly diversifying groups are evolving plumage color faster. Since the inception of the Great Speciator paradox, this relationship has been assumed, but it is for the first time, to my knowledge, that they have been empirically shown to be linked. So what is driving this rapid color evolution? Measures of whole body sexual dichromatism for both color and brightness were not supported as a predictor, nor were estimates of sympatry, both in the breeding and non-breeding seasons. To assess the role of drift, we used island size as a proxy for population size. However, this was also not supported as a predictor for rapid color evolution. However, island size is just one proxy for population size. When we used heterozygosity estimates from the resequenced genomes that I discussed earlier, linear models supported a statistically significant and negative relationship between the rate of color evolution and lower values of heterozygosity. This was supported for both sexes and both color rates. These red outline dots here are estimates derived from toe pads of museum specimens, which are likely overinflated due to issues with degraded DNA. When I removed any taxa that were represented by these toe pads, this pattern held with only half of the diversity still represented. What this suggests is that smaller population sizes evolve color at faster rates, thus suggesting that neutral mechanisms are contributing to rapid phenotypic evolution. So to wrap this up, we've shown that the rapid pace of speciation in this genus of great speciators has contributed to high ILS, but that these lineages are not completely genetically isolated owing to evidence of gene flow. We found that the fastest diversifying lineages have intermediate values of dispersal ability, providing the first empirical support to this hypothesized relationship in great speciators. We also showed this pattern is being obscured by predictable trends towards flightlessness in volant island birds. And finally, we showed that faster diversifying lineages have faster rates of phenotypic evolution. We also show that there's a negative relationship between rapid color evolution and heterozygosity, suggesting that genetic drift is a mechanism for rapid phenotypic evolution in this genus of great speciators. I'd like to end by acknowledging that this work would not be possible without the countless collectors since the 1880s that have collected, preserved, and provided museum specimens for research like ours. I'd also like to acknowledge our funding sources and the many people that have helped with this research during my PhD. So just to note, I will also be presenting this research at the in-person conference next month. If you'd like to talk more then, please reach out. And now I think there's time for questions, for 1 minute of questions.
Yep, and we've got a couple of questions in the chat. And so the first one is about whether you've considered hard polytomies in those places where you're getting nodes that are almost 33 1/3 for each possible resolution.
Yeah, I didn't formally test that. I don't know. I haven't really looked at that. I think they just are so rapid during their evolutionary history that it sure feels like it, for sure.
And one last question from the chat, which is whether there's a relationship between size and inhabiting remote islands.
So no, because our smallest birds weren't just in the most remote islands. There are some that were quite small. I technically haven't tested it specifically. I would like to have had the mass data for all of these birds, but the overwhelming majority of them were collected in the 1920s during the Whitney South Seas expedition. So unfortunately, no. And then also if we use normal species estimates of masses, it's kind of undercutting the subspecies level of diversity that this project includes. So.
Okay, thank you very much. With that, we will conclude the Ernst Mayr Symposium. Really fantastic set of talks. You can have virtual applause for everyone, for all of our speakers. And there's no more Ernst Mayr talks, but starting again at 6 in an hour, there's another final session of the virtual conference. So have a good evening and hope to see some people online later. Thank you all for coming. And thanks to our presenters.